Hello and welcome back to cell membranes. We are officially in the functional side of cell membranes. We just learned about the structure. Remember that the structure of a cell membrane is a fluid mosaic model made of three components. You got your phospholipids, which is the fluid part. You got the mosaic part, which is made of proteins and cholesterol, and each of them has a specific role inside the cell. Now we're going to move on to the function, and I'm just going to give you a brief definition of the function, but then I'll give you examples of what that looks like coming up. So, big letter A under cell membranes was structure, so big letter B is going to be function. And we'll just give the brief definition of what the function of a cell membrane is. So, what the function of your cell membranes is to provide selective permeability to control transport in and out of the cell, that's the main function, and to maintain cell shape and attachment to other cells. So if I'm a tissue, I actually can't just be a lone cell walking around by myself. I actually need to be attached to the cell next to me so we can create this large tissue. Some cells don't attach, like blood cells. They need to be free-flowing. So depending on what kind of cell you are, you need that job. So the function of a cell is to provide selective permeability to molecules across, in and out, to molecules in and out of the membrane. To provide cell shape and to provide cell attachment. So transport, shape, and attachment. Those are the three big functions of your cell membrane. What we're going to focus on right now is the transport part, the selective permeability part. So if I'm permeable to something, that means I'm letting something in or out of me. So if I'm selectively permeable, I'm only letting certain things in and out that I want to. Depending on the type of cell, it could be anything from calcium ions if I'm a muscle cell to, I don't know, sodium ions if I'm more of a cardiac muscle cell. So it just depends on what's going on. So let's talk about the two different types of transport within a cell membrane. Two different types of cell membrane transport. The first one we're going to talk about is passive transport. Let's define that, and then we'll give a couple examples of what passive transport is. So, I've told this to my students in the past, if somebody is passive aggressive, instead of telling you directly, here's what I need you to do, or here's what I was thinking, I kind of skirt around it and I'm passively addressing it and not telling it to you directly. By being passive, I'm not using any energy and trying to get my thoughts across to you. So if I'm passive, if I'm transporting something passively across the cell membrane, I'm essentially transporting without the use of energy. And in order for that to happen, it means that what's the passive transport, it's actually going down a gradient. And so let's define this, and then we'll define it in a second. So passive transport is transport across the cell membrane without the use of energy. Across the membrane without the use of energy. And this type of transport usually goes down a concentration gradient, and we're going to talk about that right now. But first I want to erase the board. So really quickly, we're going to focus on passive transport for a little while. So the two different types of cell membrane, the first one is passive. transport across the cell membrane without the use of energy. Now there's two types of passive transport. So let me just rewrite number 1 up here. So we know where we are in our notes. Okay, so passive transport. The first type of passive transport I want to talk to you guys about is diffusion. So if you've ever, have you ever had a moment where someone sprayed air freshener in a house or perfume in a house, and they sprayed it in one side of the house, and eventually you smell it somewhere else? That's because those particles of air freshener or perfume or whatever it was eventually diffused from an area of high concentration of where it was sprayed to an area of low concentration to where there was none. So that's called diffusion, when you go from something where you have a whole bunch of something to a whole lot of nothing. It always goes from high to low. That's what we call a gradient. So let's define passive transport. Well, we already did passive transport. Let's define diffusion. This is the first type of passive transport. So diffusion is the tendency of molecules to spread from a region of high concentration to low concentration. So diffusion, the tendency of molecules to spread from a region of high concentration to low concentration. Okay. And what that is, when you have a high concentration to a low concentration, that region right there, that's called a gradient. So if I were to draw a gradient, like I would have high concentration of whatever substance this is, it would just slowly diffuse down to the low concentration. You have something high going to low. That's what a gradient is. Now if you think about it in a cell membrane, you can see how things naturally just want to go from an area of high concentration to low concentration. But not everything gets to do that in a cell membrane. Remember what the cell membrane is made of. You have your phospholipids, your proteins, and your cholesterol. Okay, let's focus on the phospholipids. They have that hydrophobic core going along the middle. So if I am hydrophilic, if I'm a hydrophilic molecule, can I get through that hydrophobic core easily? I can't. So not everything gets to pass through via diffusion in a cell membrane. Only a few things can. And in order to pass through with diffusion in the cell membrane, you have to have these qualities to you. You got to be small, you got to be hydrophobic, and that's it. You got to be small and hydrophobic. So not many things can actually do this. So only small, hydrophobic. Another word for that is nonpolar. So if you're a polar, you're water, yeah? If you're nonpolar, you don't have that ability like water does. So small hydrophobic molecules can pass freely, can diffuse, I should say. Freely across the membrane. Here's a couple examples. Example. I want you to think before I write something out. So if you're an organism and you're living, you're breathing in what gas every day and expelling what gas every day? You're taking in oxygen and you're releasing CO2, carbon dioxide. You want those gases to be able to do what they need to do because every cell in your body requires oxygen to survive. And as your cells are working, they're producing carbon dioxide, and we need to get that carbon dioxide out. So we need those gases to actually pass freely across the membrane, and thankfully they do. So great example is oxygen, O2, and CO2. Carbon dioxide. If you look at the structures of oxygen and carbon dioxide, oxygen looks like this, and carbon dioxide looks like that. These are hydrophobic, they're non-polar. Compare that to the structure of water, like that. So these guys are non-polar. Also hydrophobic. When you're non-polar, you don't like to interact with water because water has such polarity to it. It has this unevenness to it. It likes to hang out with everyone else that is uneven like itself. It's not an even pulling on that central oxygen. It's pulling those hydrogens towards itself, and it's actually being polarized. Whereas oxygen, there's an oxygen here and an oxygen here. They're pulling equally on themselves, and nobody's moving in a way, if you want to think of it that way. Carbon dioxide, you have your carbon, you got your oxygen and your oxygen. They're equally pulling, so carbon is staying still. They're not polarized, therefore they're non-polar. Hope that makes sense. So, and it's great that these guys can freely pass that hydrophobic core. That phospholipid, or the head on the phospholipid, it's hydrophilic, but the fluidity of the phospholipid bilayer is allowing these gases to go right through and not be affected by that hydrophobic core. And since they're moving around, they can wiggle in between the heads, which is lovely. So only small hydrophobic or nonpolar molecules can diffuse freely across the membrane. And they're always going to diffuse. So if I have high O2 on the outside of my cell and low O2 on the inside of my cell, oxygen is going to diffuse in. That's my gradient. I have a high to low gradient. Vice versa, if I have high CO2 on the inside, let's see, and low CO2 on the outside, CO2 is going to diffuse out of my cell. That's something that you would see in your veins and your arteries and the capillary beds where the two of them like to meet. That's when you get that gas exchange going on. And that is, the reason we breathe. And when we get to this later, that's the reason inside every single cell you have this organelle called a mitochondria that does all this lovely work. We're going to get to that soon. So, not now though. Gotta wait for it. Alright, so diffusion, the tendency of molecules to spread from a region of high concentration to low concentration, to go down their gradient, if you will. Only small hydrophobic molecules can diffuse freely across the membrane, such as oxygen, carbon dioxide. And this is where you gotta be careful because carbon monoxide, stuff like that, those are also hydrophobic as well. So some toxic gases can pass freely into your cells, and that's why they're so scary sometimes. If you have a carbon monoxide monitor at your home, or even like methane, stuff like that, certain gases are very hydrophobic. They're non-polar in their shape, so they can easily pass through the membrane. But they gotta be small. So when we're talking like, 3, 4 atoms, it's really not that big. They could do it. So oxygen and carbon dioxide, good. Keep those going. So that's the first type of passive transport. The second type of passive transport is going to involve water, because our sweet cells, there's a lot of water on the inside and there's water on the outside. So we got to make sure that we keep that balance of things going well between the cells. So, and I'm wondering, if you're wondering, hey, I thought you said passive transport is, diffusion. You're going down the membrane, the concentration gradient in the membrane, and water is polar, it's hydrophilic, by definition it loves itself. How can it pass through that hydrophobic core? Great question. Wait for it, we're going to get to it right now. So, this is going to be little b, osmosis. You've heard the words osmosis before, like when something just kind of seeps into something. That's exactly what's going on here. So osmosis is diffusion of water across the membrane, but it does something a little different. water can't go across the membrane. It has to use a protein to allow it to go down or into the cell or out of the cell. So osmosis is transport of water across the membrane. And there's a few things we've got to talk about with this. Since water is hydrophilic, it loves itself, it's polar, it does all that. Since water is hydrophilic, it can't just freely cross the membrane. It has to use a protein. And this is called it's a transport protein. And since we're using a protein to do passive transport, we're not using any energy to move something from one side to the other. We'll do that later. Since we're not using any energy, this is called facilitated diffusion. You are actually facilitating the diffusion of a substance across the membrane. So facilitated diffusion is essentially the use of a membrane protein to do passive transport. The use of a membrane protein to do passive transport. And now specifically for water, water uses a special protein called an aquaporin. So aquaporin proteins transport water. So if you're looking at your typical cell, and I'll make a little channel here, little channel here, and these are all aquaporins. I'm pretending to make them all. So these little guys are aquaporins. They're going to let water in and out of the cell as needed. And you may be asking yourself now, how does water know when to go in or out of the cell? That is a great question. Let's talk about that. So osmosis, it uses facilitated diffusion. Here's how water knows how to move. Water will essentially move to the region that has a high solute concentration. A solute is a substance that is trying to be dissolved in water, right? So if you have a whole lot of it, water is going to come and try to dissolve you, essentially. So water moves in the direction of high solute concentration. High solute, you could also think substance. That's another way to think about it, of high solute concentration. Okay, we're going to have a whole bunch of examples on how this works. So water moves in the direction of high solute concentration. Now I'm going to erase this part and do a little number 3. We have a word for this, for us, what makes water move in the direction it needs to move. It's called tonicity. Tonicity. The word tonicity means this is the ability of a solution to cause water, or to cause a cell to gain or lose water. So if I'm a cell and I have a specific tonicity, let's say I have a very high solute or substance concentration inside my cell, water is going to want to move in. All right? We're going to talk about all these examples. So tonicity is the ability of a solution to cause a cell to gain or lose water. The ability of a solution to cause a cell to gain or lose water. And there's 3 types of tonicity out there. I'll just do little one. Try to get my notes nice for you. First thing you have is a hypotonic solution. So hypotonic. A hypotonic solution means that what's going on is you're looking at the solution inside the cell when you do these things. So if I am hypotonic, that means, actually it's a solution the cell is living in, I should say. So if I'm hypotonic, I'm a cell and I'm floating around in the solution, what's around me? It means that there's less solute on the outside than there is on the inside. So hypotonic has a low solute concentration on the outside of the cell compared to the inside. Okay. If I'm hypotonic, I have not as much solute on the outside than I do on my inside. So you got to pretend you're the cell looking out. What solution are you floating in right now? If I am isotonic, the solution concentration is equal on the inside and the outside. It's good balance. So solution, actually I'm going to say solute, forgive me. Solute concentration is equal inside and out. So you got hypo, you got iso. If you know enough science going on in your background, the last one is hypertonic. Hyper, whenever you hear the word hyper, it means too much of something or it's elevated. So opposite, if hypotonic is low solute concentration on the inside, hypertonic means there is high solute concentration on the outside of the cell. So high solute concentration on the outside of the cell. Okay, I'm actually going to stop here because I want to leave time, be respectful of your time on the videos. But when we come back, I'm going to show you examples of what a hypotonic, isotonic, and a hypertonic cell looks like in solution and compare what a plant cell versus an animal cell would look like. And then you'll be able to recognize, they're in hypertonic, they're in hypertonic, blah, blah, blah. You're going to figure it all out. So come back, we'll do an example, and then we'll move past passive transport and get to the other type of transport as well. Thanks.